Bismillahirrahmanirrahim. Last time we have finished up to the histology of esophagus. Now we move on to the next organ, stomach. Now this slide is showing you the anatomical position of the stomach as well as the various functional aspect of that organ. Now this is the esophageal end from where esophagus open into the upper part of the stomach, the fundus part, then the body, then is the pyloric region, and this is the pylorus sphincter. From here the duodenum starts. Two curvature, lesser curvature and greater curvature. Now the stomach, food starts to be digested and absorbed. What are the performance function? Then the food start to be digested and absorbed. Over here, absorption is mostly limited to water, alcohol, and some drugs. Very limited absorption in this region of the GIT. Food can stay in the stomach for 2 hours or more, broken down chemically by gastric juice and mechanically by contraction of smooth muscle. So, dual function of stomach, that it mechanically broken down into multiple small pieces with addition of gastric acid as well as pepsin, so food becomes semi-solid. This is again a mechanical function, but the chemical function is addition of gastric acid as well as the enzyme pepsin with the lipase as well. The broken part or the broken up food at this end of this process is called a chyme. No semi-solid broken part of the food incoming from the esophagus, no change into the chyme, is a semi-solid liquid form of a food. Now, what are the functional aspects? Both exocrine and endocrine organ. ये आपके लिए एक नई बात होगी, एक्सोक्राइन एंड एंडोक्राइन ऑर्गन. आपके जहन में फौरन जाएगा कि एक्सोक्राइन है तो उसकी डक्ट सिस्टम किधर है, एंडोक्राइन है तो कौन से इसके एंडोक्राइन सेल हैं. We will discuss in coming slides. Digestion of carbohydrate by salivary amylase. Over here, because of the salivary amylase from the salivary gland is mixed up in the food particle, so partially broken down of carbohydrate into simpler compound in this region. Initial digestion of protein by pepsin, because pepsin is an enzyme which is concerned for the protein digestion. Then the mechanical conversion of bolus into chyme. So these are the major function of the stomach. Now, the histology of the stomach will start with the transitional between the esophagus and the stomach. Now, in this diagram, you see very much clear about this. This is the stratified squamous non-keratinoid epithelium. Over here, it is the typical epithelium of the esophagus. Now, from there, you see the epithelium is going to change to the simple columnar epithelium with depression within the lamina propria. This is called as a gastric pits. The gastric pits starts when appearance in the transition in the cardiac end of the stomach, from where the esophagus open into the upper part of the stomach. So this transition of the epithelium from stratified squamous up to the simple columnar with gastric gland. So gastric gland starts appearing over. Although muscular mucosa is the same, lamina propria is the same, submucosa is also visible in both the slide. So the changes usually present between the esophagus and the stomach is in the mostly the mucosa. So this slide will tell you the transition between the esophagus and the stomach. Now, as usual, the histology of the stomach can be discussed. Mucosa, submucosa, muscle layer, and serosa. So we'll start with the mucosa. Mucosa is thrown up into fold called rugae, visible at rest. Now, what are the rugae? Rugae is the visible fold you can see by naked eye. When you open the stomach, you see a multiple fold at rest, fold of the mucosa and submucosa. This is also called as rugae formation. Epithelium and lamina propria dips in form of a gastric pits. The mucosa is thrown, mucosa mean the surface epithelium, lamina propria muscularis mucosae, that is dipped inside to form the gastric pits, elongated gastric pits. Lamina propria consists of loose urethral tissue with lymphoid collection. Lining epithelium is simple columnar. Beneath lamina propria is a muscularis mucosae. So in the lamina propria, the epithelium is thrown into gastric pits lining by the gastric glands. So the gastric glands are the modification of epithelial tissue in the gastric mucosa which dips into the lamina propria to form the gastric pits. In this diagram, you see, so this is the simple columnar epithelium, lamina propria. When it dips down to form multiple glandular tissue, these are the gastric pits elongated. So these gastric pits are lining by a modified epithelial cell which are secretory in nature. These are called a gastric gland. Rest is the lamina propria is having all the elements containing the connective tissue, loose urethral tissue. Then is the blood vessel, lymphoid tissue is also present. Epithelium is simple columnar. Muscularis mucosae is also visible in this diagram. In this HNE stain, this is the muscularis mucosae layer, simple squamous epithelium. Now, over here we will discuss the gastric glands. These are the modified branch tubular gland lie in the lamina propria, divided into three parts. Because these are elongated, ये लंबे लंबे होते हैं, having starting from the luminal side. Luminal side से start होंगे, dip down in the lamina propria up to the muscularis mucosae. So it can be divided into three parts: apex, neck, and base. The apex, neck, and base. Apex is superficially toward the luminal side. The neck is the narrow part, and the base is body or base is toward the lamina propria. It is elongated funnel shape, so it is the base. Now, this gastric gland contains following types of cell which have their specific function to perform during digestion. The first is the mucous neck cell, second is the oxyntic parietal cell, then is the third is the chief zymogen cell, then is the enteroendocrine cell, then the stem cell, fifth, sixth is the D cell, and seven is the G cell, gastric releasing G cell. Now we discuss one by one, briefly, all these cells in the gastric gland. Now, in this picture, it is the broad description of all types of cells which are located within the gastric gland and their function. Now, this is the luminal side or gastric pit apex. This is the elongation of the gastric gland, bronch tubular gland moving toward inside in the lamina propria. So look at the shape, it is a inverted U-shaped gastric gland. Now, this gastric gland is the exocrine part of the stomach. It is the exocrine part of the gland of the stomach. Now, always remember that it is just because it is a well-defined duct over here, single duct, pouring the secretion within the lumen of the gastric cavity. So it is the exocrine part of the gastric gland. Now, in this case, you see this is the narrow neck and this is the base or body. This is the base. Sometimes it is also called as a neck, body, and a base. So base lies deep in the lamina propria. The elongated part in the middle is called the body, whereas the apex is toward the luminal side, where it opens the secretion within the lumen. Now, important cells, the mucous neck cell, because these are located along the neck or upper part close to the luminal side. These mucous neck cells are columnar cell, and they, as the name indicates, it is concerned for the secretion of mucus, protect lining, and also secrete the bicarbonate ion. It is and it secretes the bicarbonate ion. Now look at the picture in this neck region and upper part of the body is the parietal cell. So these parietal cells are important because these are the cells which are D-shaped. These are the cells which are D-shaped and they are highly eosinophilic in staining. These parietal cells secrete the gastric acid intrinsic factor, and calcium ion absorption. Three functions it perform: release of intrinsic factor and gastric acid secretion and calcium ion absorption. Now, enteroendocrine cell, these are the enteroendocrine cells which are located at the margin of the body and also along with the neck of the gastric gland. These enteroendocrine cells are the endocrine part of the gastric gland. Now, this is the both exocrine and endocrine gland of the body, just as stomach. So, this enteroendocrine or enterochromaffin-like cell, they are the endocrine part and they secrete the histamine within the blood because they pour the secretion directly into the lamina propria in the blood vessel. Now, what about the chief cell? Chief cells are the cells which are protein synthesizing and they are located within the body and also the close to the base are all around are located. So these are the cells which secrete the pepsin and gastric lipase, two enzymes, pepsin and gastric lipase secreted by the chief cell, and these cells are highly basophilic in staining. These cells are highly basophilic because they are the protein synthesizing cell. Now again the D cell and G cell, these are the endocrine cell because they pour their hormones, somatostatin and gastrin within the blood. So they are also present within the base and body lower part, and they secrete the somatostatin which inhibit the acid, gastrin which simulate the acid. They are also present within the gastric gland. Now this is the brief description of location along with their function of various types of the cells present within the gastric gland. Now one by one, histology of all these cells. The mucous neck cell. Look at the picture. This is the histological stain of the enlarged high power, the H&E stain. Look at the picture. The nuclei are toward the basal side, elongated, the apical zone and full of mucinous granules. Mucinous with microvilli are also present, but they pour the secretion directly within the lumen. So secretion first accumulate in the apical zone of the cell and then they pour within the lumen. So it is nullifying the acid present within the gastric mucosa. So M cell, they are present mostly in the neck region, basal zone nucleus, then the apical zone is full of mucinous granule. Look at this picture, which is hypothetical diagram. Macrovilli are there, apical cytoplasm is full of mucinous granule, Golgi apparatus is supranuclear, mitochondria are there, the rough endoplasmic reticulum as well as smooth septum both will be present, but the feature prominent is that is the mucinous granules in the apical zone. Location is in the neck region. Function is it secretes the bicarbonate as well as the mucin to buffer the acid present in the gastric mucosa. Now this is the mucous neck cell the biochemistry. Look at the three phases of the pH of the gastric mucosa. In the luminal side, more H ion, so pH is much more reduced, it is over 1.5. And when it moves toward the cellular region, because more carbohydrates are releasing from these mucous neck cell, so they will nullify. The pH become 2 at the apical zone, more toward the cellular side become pH 7. So at the cell level, close to the apical zone of the cell, the pH is more toward alkaline, but the apical zone within the stomach cavity, the pH is acidic 1.5. So this is the three zone: H ion zone, bicarbonate zone, and these are the basal zone for absorption and secretion of H ion and bicarbonate. So three zone can be biochemically divided in the gastric mucosa and especially the mucous neck cell. H zone, that is absorption and secretion of the H ion and bicarbonate, and then the zone of secretion where H ion and carbohydrate then form the carbonic acid with the carbonic anhydrase, release of HO3 from where the pH is 7 at that. But when the H ions are proton pump will occur, the H ions are protrude or H ions are secreted within the lumen, so pH suddenly change to form the 1.5 pH, with the chlorine coming from the basal side from the HCl. So this is the pH level at different status of the mucous neck cell within the lumen of the gastric mucosa. Now look at the picture. This is the oxyntic cell, mean it is the parietal cell. Next cell is the parietal cell, which is highly acidophilic cell, as the picture is denoting you that the nucleus is almost toward the middle side, more toward the middle side, and the apical zone is having the intercanaliculi of the cytoplasm, and the nuclei are also visible. The cytoplasm is highly acidophilic. So this is the oxyntic cell, which secrete the HCl as well as the intrinsic factor and calcium ion absorption. This is the hypothetical diagram in which you see, these are the imagination of plasma membrane to increase the surface area. This is the typical picture of a ion secreting cell. Always remember, the ion secreting cells are typically that they have the intracellular canaliculi. Now this is the intracellular canaliculi, imagination of the plasma membrane within the cytoplasm to increase the surface area. More absorption, more release, more activity of the cell with the help of mitochondria. So more production of iron within the lumen is the functional aspect of parietal cell. Now look at the picture. In this case, mitochondria are abundant in these canaliculi. The nucleus is present. The endoplasmic reticulum is mostly smooth, but because it is releasing the intrinsic factor also, so there are both smooth as well as the rough surface. Endoplasmic reticulum, Golgi apparatus are present, and then the lysosomes are also present. The characteristic feature is intracellular canaliculi to increase the surface area, more mitochondria. So that is why the staining is eosinophilic. So parietal cell is D-shaped. It is wedge-shaped cell. Location is neck region and upper part of the body. This is the biochemistry of the parietal cell. Now in this case, the carbon dioxide in the water with the help of carbonic anhydrase is converted into the HO3 ion. HO3 ion is released within the blood. Then the H ions are, with the help of proton pump, is poured within the lumen of the gastric mucosa with the help of ATP molecule, because ATP is converted into ADP, providing the energy for the proton pump. So H ions are poured according to the against the concentration gradient of the H ion poured within the lumen. So we need energy over here. So this energy is mediated by the ATP molecule. So this mechanism is called as proton pump. So carbonic anhydrase convert the H2O to carbon dioxide. So production of HO3. Histamine and gastrin having an influence of these parietal cell. Now look at the picture of the chief cell. The chief cell, as the staining is indicating, it is a highly basophilic in type, because its nuclei are pushed toward the basal side. The cytoplasm is containing the zymogen granules in the apical zone. Macrovilli are there. The endoplasmic reticulum is rough because of the protein synthesis. So nucleus is having a well-defined nucleolus. Look at the picture. These are the nucleoli, well-defined nucleoli, because these are the characteristic feature of production of ribosomal RNA. So it is a feature of protein synthesizing cell. Now this is the diagrammatic presentation. Apical zone is full of zymogen granule. The luminal side and macrovilli. The nucleus is toward more basal side. The endoplasmic reticulum is rough. Golgi supranuclear Golgi complex. Multiple vesicle granules of zymogen granule, because these are proteinous granule. These are precursor of pepsin enzyme. Now difference you will have to remember. The location is neck, base, and the body of the gastric gland. They secrete the pepsin, and which is concerned for the degradation of the protein molecule. Now this is the comparison of both chief cell and parietal cell. Quite obvious. Now look at the picture. Nucleus basal side. Nucleus is almost in the center. Apical zone is granules full of. These granules are of zymogen granule. Staining is the basophilic, staining is the eosinophilic, intercellular canaliculi is the characteristic feature of parietal cell. No canaliculi formation. The rough endoplasmic reticulum is more prominent in the chief cell, where it is less in the parietal cell. The mitochondria are prominent, more abundant, mitochondria are relatively less. But this is characteristic feature. This is the proteins synthesizing cell, this is the ion transporting cell. Difference is quite obvious. Intercellular canaliculi to increase the surface area, whereas the luminal side of granules is the characteristic feature of the protein synthesizing cell. Now, enteroendocrine cell, this is again a cell which is concerned for the excretion of this is the endocrine part of the gastric mucosa, found in the stomach and usually seen in the electron microscopic picture. So the nucleus is more toward the supranuclear, toward the apical zone. The basal is broad. Over here the secretions granules are present on the basal side, because the secretions are directly poured within the lamina propria in the blood vessel. So there is no secretions pouring within the lumen of the gastric pits. So the secretions are poured within toward the lamina propria, where they are absorbed in the blood vessel. Basolateral domain macular project into the lumen, but maybe some granules may be poured within the lumen, but mostly the secretions are poured in the lamina propria, where they are get absorbed. So they secrete the somatostatin, histamine, and very other vasoactive amine within the blood vessel to coordinate or to stimulate or depress the activity of the stomach during the digestion. So this is the important part of the endocrine portion of the gastric mucosa. Now this is the diagrammatic presentational table of the various types of hormone, their site of secretion, production stimulus, target organ and action. For example, gastrin, stomach mucosa, mainly G cell of the pyloric antrum, presence of peptide and amino acids in the stomach, ileocecal valve, relax valve. So these are the various functions. Promote intestinal muscle contraction. So all these functions of the gastric ghrelin, stomach mucosa mainly fundus, fasting state level increase prior to meals, hypothalamus, target organ, regulate food intake primarily by the stimulating hunger and satiety center. So this chart is for academic interest. You will have to remember it with the serotonin. For example, these are the neuroendocrine cell present within the gastric mucosa, histamine, serotonin, somatostatin. So all these are the cells which are concerned for the release of ghrelin, G cell. So they invariably affect the activity of the GIT by stimulating higher center or locally. So this, for academic interest, you will have to memorize this also. Now, what about the submucosa? Now, submucosa in this diagram, you see it is the loose or dense connective tissue. Muscularis mucosae are also present. The mucosa contain the blood vessel, nerve plexus also. So this is a characteristic feature of the stomach as well. But always remember, the gastric glands are present within the mucosa, not the submucosa, within the lamina propria. Muscularis externa, so these are the three layers visible, that is outer longitudinal, circularly arranged, again outer longitudinally arranged. So three layers of the outer longitudinal muscle, inner circular smooth muscle, muscularis externa. This is a characteristic feature, but always remember, this is the smooth muscle, not the skeletal. It is smooth muscle controlling by the involuntary activity. Now the last one is the serosa. You see in the visceral peritoneum is the last one. So this is the lining epithelium, and then is the connective tissue layer, longitudinal muscle fiber, oblique muscle fiber, circularly arranged muscle fiber, blood vessel in the gastric mucosa, submucosa. These are gastric gland. This is the beautiful picture of demonstrating all the layer of the serosa present, all the layers of the gastric mucosa along with the gastric histology. But over here, the outermost covering is the visceral peritoneum, that is the lining epithelium along with the underlying connective tissue. Now, the comparative histology of the region wise of the stomach: cardiac region, pyloric region. Structure: gastric pits, short pits, long pits. In the pyloric region, the gastric pits are elongated, whereas in the cardiac region it is short pit. Gastric glands are long tubular gland, where the short called branch tubular gland. Gland cytology: all cell lineage, few parietal cell with more mucous cell. G cell and D cells for gastric and somatostatin hormones are more prominent. So in the cardiac region, all cell lineage will be visible, all types of cell. But pyloric region, more specifically, few parietal cell, mucous cell, G cell, D cell, gastric, somatostatin hormone become prominent. Now, inshallah, next we discuss with the small intestine. Thank you very much.